So good evening, everyone. Welcome to Cell Talk presentation. For today's presentation, Vijay is a presenter and he is going to present on the topic predicting prostate cancer molecular subtype with deep learning on histopathologic images. Before the presentation, I would like to introduce Vijay. Vijay Padul is working as scientist bioinformatics at Renix Life Sciences. At Renix, he is leading multiple research projects on bioinformatics, genomics, and next-generation sequencing research-related activities in the organization. His current research projects include the development of personalized neoantigen cancer vaccines and the utilization of publicly available multi-omics cancer datasets to create new insights into cancer biology and its application in cancer therapy. Vijay Badul has completed his PhD in life sciences from TMC AC TREC Homi Bhabha National Institute, Navi, Mumbai. His PhD study focused on cancer genomics involving multi-omics analysis of glioma brain tumor subtypes. At Renix, Vijay has established bioinformatics pipelines for personalized cancer genomic analysis, and his research has highlighted the importance of molecular marker analysis in personalized cancer therapeutics to match the treatment strategies to patient cancer tissue molecular characteristics. His contribution include assessment of patient HLA genotype status for tailoring the personalized neoantigen peptide therapy and glioblastoma subtype analysis. His larger aim is to utilize personalized genomics approaches for aiding effective personalized treatment strategies for cancer patients. I welcome Vijay to start with his presentation.
Thank you, Uma, for the introduction. Hello, everyone. For today's Cell Talk presentation, I have chosen a research article predicting prostate cancer molecular subtype with deep learning on histopathologic images. The original authors are Erica et al. Title for the presentation is Harnessing Advanced Artificial Intelligence for Improved Cancer Diagnostics. The topic selected for presentation is Artificial Intelligence in Cancer. The paper was published in the journal Modern Pathology with impact factor 7.1 in the year 2023. This research has impact on prostate cancer molecular diagnosis using immunohistochemistry and precision medicine. So prostate cancer is highly prevalent in male, and this is the second most commonly diagnosed solid tumor in the United States. Therefore, this tumor, it has high significance for development of deep learning or artificial intelligence mediated pathological diagnostic aids so that accurate diagnosis can take place of these patients. And also molecular grading and advanced stages of these tumors can be identified. And also there are attempts to identify what are the molecular alterations, key molecular alterations that are affecting different prognostic implications of the cancer patients, and also which could be predictive for therapy response so that particular therapy can be selected for that particular cancer type. And also these identification of these molecular subtypes is also important to identify associated risks for with respect to the germline variations found in those particular cancers. So therefore, in that respect, identifying molecular subset of that prostate cancer is essential for the prognostic implications that hold for the cancer patient. Now, majorly in prostate cancer, these two types of molecular alterations are found. One is PTEN loss, and second is ERG gene fusions. PTEN is a tumor suppressor genes, and it's highly prognostic in nature. If this particular genetic alteration is present, then it is predictive of metastasis of that particular patient, and also particular therapeutic approaches are also available if PTEN loss is present or if it is absent. The second common alteration is ERG gene fusion. ERG is a gene which is classified as an oncogene, and because of these gene fusions, the cancer development and also progression occurs. Other pathogenic mutations, these are found in DNA repair genes, and these repair genes, these include mismatch repair, function of the cell, and also homologous recombination, which are essential for maintaining the integrity of the genome. The patients which have these mutations in the repair pathways, these are eligible for PARP inhibitor or immune checkpoint inhibition based therapeutics. And As you see, these are different kinds of different molecular alterations that are found. So it is crucial to identify tumors which hold these kinds of alterations as early as possible, so that treatments can be made. And newly advancements in the deep learning and also artificial intelligence, it can be harnessed for this particular aim. So previously in renal cell carcinoma, they have shown a very strong correlation between tumor histologic subclass in the clear cell renal cell carcinoma, which are like VHL gene inactivation, etc., and the molecular alteration status and the histologic subclass of that particular cancer. They have very strong correlation. So can this also be established in the prostate cancer also? And if this can be established, this will be immensely helpful to the patient classification and also for the treatment selection of the patients. So for this particular aim, this authors in this particular paper, they have attempted to use the image classification and also image processing using artificial intelligence to identify molecular subgroups in this prostate cancer type. So for this, they have used whole slide images from thousands of different prostate cancer patient tumor histological slides, which are H&E stained slides, and used their custom-made artificial intelligence algorithms, deep learning algorithms, to identify molecular subtype association with the histologic features on those slides. The present challenges in this particular field, they include that for identification of these molecular alterations, as we have previously seen in the earlier slides, genomic sequencing is the best method, and this is the foolproof method. That means if in genome sequencing that particular mutation arises, that means it is definitely present there. Histopathological diagnosis comes second, but specific mutations require specific methodologies for immunohistological detection. So these are not possible. And together that genomic sequences still remains expensive in many parts of the globe. Even if it's expensive sequencing has become affordable in many countries, but still these are out of reach in many parts of the country, in the different parts of the globe. Apart from that, this technique remains time-consuming. There are multiple steps, and there is a high possibility that sample choice that is used can also affect the final result that is available for analysis. Then the commonly used approach is use of trained histopathologist eyes to identify sections of these tumors, which are H&E stained and different immunohistochemical stained, and to identify what molecular subtype is existing and how much of this particular tumor is having cancerous tissue in it. Then another disadvantage for this method is that such highly trained histopathologists, they are few in number, and therefore this two-pronged challenge is there, that there should be an alternative for genomic sequencing, which could be cheaper, and also highly trained histopathology requirement would not be required for that particular method. So here is where this research group is trying to present their algorithms for computational detection of subclass of prostate cancer. So objective of this research was to develop deep learning algorithms-based methods to screen for genomic alterations using diagnostic H&E stain slides to provide an inexpensive test to rapidly triage patients who could most benefit from germline or somatic sequencing early in the disease progression. So in this research, they have used hundreds and thousands of tissue slides from different cohorts. One of them being the natural history cohort with 288 cases with ERG fusion known status. Then the case cohort with 248 tumors with previously published ERG fusion status. The race cohort with 375 cases with previously published ERG fusion status by immunohistochemistry. They use this whole slide images and then this scan using Hamamatsu S360 digital slide scanner at 40x magnification and also at 20x magnifications. They studied these slides in two different groups with known molecular alterations, that is ERG fusions and PTEN. And these molecular alterations were already established by using immunohistochemical staging. The ERG gene fusion were detected by FISH, and PTEN immunohistochemistry specific was used for identifying PTEN gene deletions. So here is the overview of methodology used in this deep learning framework. So here they use the slides for identifying regions where tumor is present. Then out of that tumor, they recognize features for deep learning. The feature extraction and classification is done, and explainative map is generated to identify why did this particular algorithm choose those particular features for that particular class. And as you can see in this particular stage, this base slides, these white areas, they represent the removed tissues, that is called as tissue punches. The tumor is not there, so whatever area that is visible in the pink shade, that is the tumor area. And out of that, the specifically bluish tinged area, which is generated by the algorithm, this is where they have detected the molecular alterations by using their specific algorithm. So here this slide describes their algorithm. So starting from the tissue slide, they extract 4296 by 4096 images, and they create a matrix and a tensor out of these images. Then out of that images, they subclassify or subset them into 256 patches of 256 by 256 pixel size. And after that, they use their algorithm for CLS, VITS, and after this extraction of the features, then they create a tensor and use their developed algorithms to identify which particular area having tentatively that particular predicted molecular alterations. So here also, once they detect that particular molecular alterations in their previous program, then they again use their algorithm to identify those specific areas in which that molecular alteration is found to be present. So this is a very molecularly heterogeneous tissue slides. That means these mutations may be present in some areas and may be absent in some other areas. So therefore, you can see that the blue color is found to be present in some areas, where in other areas it is still pink, that is showing the normal tissue region. So this kind of identification is being done. So here it is comparison between a human trained histopathologists identification of the tissue section, and here it is the sections that has been identified by deep learning algorithm. So pretty much a lot of overlap is there if you see the blue regions and the pink regions, but the specificity of identification by deep learning method is very high in this B slide as compared to the histopathologist. So that shows that the correlation of histopathologist human trained eye identification also correlates with the deep learning based identifications, but deep learning based identification is giving better results as compared to the histopathologists. So here the correlation for PTEN loss by histopathologist and PTEN loss by deep learning. So it is showing positive correlation with a p-value of 0.097. So this is the comparison of the AUC curves for different cohorts that was used with these deep learning algorithms. So natural history cohort with radical prostatectomy. So AUC is 0.91, which is the highest among these different cohorts. Then there is case cohort with 0.86, race cohort with 0.89. Then for needle biopsy, radiation therapy cohort is 0.78, and active surveillance cohort it is 0.80. Then for the earlier slide, it is showing the algorithm accuracy for ERG fusion genes, and in this particular cohort, it is showing the accuracies for PTEN related molecular alteration detection. So here also molecular history cohort 0.81 for radical prostatectomy, case cohort 0.72 AUC. In race cohort 0.8 and radiation therapy cohort for needle biopsy it is 0.75. So coming to discussion. So this results shows that the machine learning algorithm, this can be applied to hematoxylin and eosin stain images of the prostate cancer to identify underlying genomic alterations. That means if the slide is provided with this particular algorithm, it can determine whether PTEN deletion is present or ERG fusion is present in those particular slides, which is kind of an alternative, not accurately, because the highest accuracy that we are seeing is 0.91. So it is not like 100% accurate, but still kind of a good estimation can be made whether that particular molecular alteration is present in the slide or not. Then by using thousands of H&E stain slides and AL algorithms, this particular rearrangements and mutations, here they have only used two particular mutations, but many more such mutations can be compared with already classified images by histopathologists and different histochemical methods, and also genomic sequencing based identification of those particular mutations and their correlated slides. That correlation can be made, and these data can be utilized or harnessed to identify the or training the model to identify specific molecular alterations. Now this study also has limitations. That limitation is that this data that is trained on the large needle biopsy cohorts. This may help to develop algorithms with improved performance in small tissue samples, but this small needle biopsy and the entire prostatectomy, these samples cannot be correlated with each other. To conclude, deep learning algorithms used in the study to predict ERG fusions and PTEN status. This proved that H&E stained images, this can be used to screen for underlying genomic alterations in the prostate cancer. The impact of the research is that with the increasing use of machine learning algorithms in daily pathology practice for tumor grading and staging, similar molecular triage algorithms could provide a quick screen for clinically actionable alterations using ubiquitously available diagnostic and H&E. So this will shorten the time period that is required from diagnosis through the genomic sequencing or again histochemistry based methodologies. These algorithms can improve the time gap between the diagnosis and treatment that is provided to the patients. This will also reduce the burden on genetic testing facilities and also counseling services, that is the trained histopathologist for this highly prevalent tumor types, and also this will improve the universal access to precision medicine. Thank you for patiently listening.
Thank you, sir, for the presentation. Audience, now the floor is open for the queries. If you have any questions, you can type in the chat box. I don't see any questions. Can you move to the last slide, please, sir? Okay, so next presentation will be by Uma Rakshita, and she will be delivering on the topic Application of Artificial Intelligence in Lung Cancer Screening, a real-world study in a Chinese physical examination population. And this presentation will be on 7th May 2025. I thank everyone for attending this session, and thank you, Vijay sir, for the presentation.